Okay, so now we move to the Krebs cycle. So Krebs cycle start with the process of link reaction. So before this, we already discussed about the link reaction. So in link reaction, pyruvate, the two molecules of pyruvate is decarboxylated and oxidized. And from this process, okay, of link reaction, it will produce two molecule of acetyl-CoA. In this cycle, acetyl-CoA, okay, so one molecule of acetyl-CoA, okay, the two carbon, dia adalah two carbon molecule, okay, so dia akan oxidize in a series of reaction, okay, occur in the matrix of mitochondria. Acetyl-CoA two carbon molecule combine with a 4 carbon molecule, okay, the 4 carbon molecule iaitu oxaloacetate, okay, OAA, to form the 6 carbon molecule citrate. Okay, so this 6 carbon molecule, this citrate, dia akan oxidize, maksudnya dia akan remove dia punya electron, okay, berlaku electron removal, and also the removal of 2 carbon dioxide. Okay, so di sini dia satu, step 3 and step 4. So 2 carbon dioxide is removed to restore, okay, to restore OAA, okay, the 4 carbon molecule untuk form again the oxaloacetate, the 4 carbon. OAA is then recycled and bind to another, okay, to the second acetyl-CoA to complete one cycle, the complete process of the Krebs cycle for one molecule of glucose. So now we look to the detailed steps in Krebs cycle. So there are eight steps in Krebs cycle. We start with the step 1 and step 2. Step 1 is the condensation process. Okay, condensation. So in this step 1, acetyl-CoA, okay, from oxidation of pyruvate, release its coenzyme A. So berlaku removal of coenzyme A di sini. Okay, and then the acetyl group of acetyl-CoA, the 2 carbon molecule ni, dia akan join with the oxaloacetate. Okay, join dengan oxaloacetate, the 4 carbon molecule, to form the 6 carbon molecule citrate. Okay, so dalam step pertama, dia akan riliskan dulu acetyl-CoA, one molecule of acetyl-CoA akan riliskan coenzyme A. Okay, so bila dia riliskan coenzyme A ni, actually ada energy yang terbebas, dan this energy is used to join the acetyl group, the 2 carbon molecule, to oxaloacetate, the 4 carbon. So dia akan bind together to form citrate. Okay, the 6 carbon molecule. Okay, step 2 is the isomerization process. Isomerization. So in this step, citrate is converted, or citrate undergo isomerization to form isocitrate. Okay, to form its isomer, isocitrate, by removal of one water molecule, the remove one water molecule, and addition of another. Okay, so ini dalam step yang kedua. Okay, step yang ketiga adalah step oxidative decarboxylation. Okay, so dalam step ni, isocitrate dia akan oxidize. So maksud oxidize, berlakunya removal of 2 hydrogen atom, and juga berlaku removal of 1 carbon dioxide. Okay, so kalau kat sini ada 1 carbon dioxide molecule. Okay, dia akan remove, okay, from the isocitrate. Seterusnya dia akan form alpha ketoglutarate, the 5 carbon molecule. Okay, So NAD plus ni dia akan terima the 2 hydrogen atom daripada isocitrate, and then NAD plus is reduced to NADH plus H plus. Okay, so ini yang berlaku dalam step yang ketiga. Okay, so for step 4 also, sekali lagi berlakunya oxidative decarboxylation. Okay, so alpha ketoglutarate, the 5 carbon molecule, undergoes oxidative decarboxylation. Okay, di mana dia akan removekan satu carbon dioxide, satu molekul of carbon dioxide, which convert alpha ketoglutarate 5 carbon into succinyl group 4 carbon. Okay, succinyl group 4 carbon molecule. And also alpha ketoglutarate ni dia akan remove 2 hydrogen atom. Okay, sebab kat sini berlaku proses oxidative decarboxylation. So yang oxidize adalah alpha ketoglutarate. So dia akan remove the 2 hydrogen atom, dan seterusnya yang akan terima the 2 hydrogen atom adalah NAD+. Okay, so NAD+ kat sini dia akan reduce to NADH plus H+. Okay, one coenzyme A ni dia akan join dengan succinyl group. Okay, dengan tadi bila dia riliskan carbon dioxide, dia akan form daripada alpha ketoglutarate 5 carbon, dia akan form succinyl group 4 carbon, dan kemudian coenzyme A akan join with the succinyl group to form succinyl coenzyme A. Okay, dan these molecules, okay, succinyl coenzyme A ni mempunyai high energy. Okay, energy dia tinggi, high energy, dan dia punya ikatan dia pun unstable. Okay. Okay, so now step yang kelima, step 5, berlakunya proses substrate level phosphorylation. Okay, substrate level phosphorylation, di mana tadi succinyl coenzyme A ini adalah molekul yang bertenaga tinggi. So the high energy bond, okay, between the coenzyme A and succinyl is cleaved, energy is released. Okay, and this energy is used to bind the inorganic phosphate. Okay, ada inorganic phosphate dalam matrix dalam mitochondrial matrix ni. So dia akan bind the phosphate tu, dia akan bind to the GDP, bind pada GDP to form GTP. Okay, then GTP akan transfer its phosphate to ADP, okay, to form ATP. Okay, and the succinyl group is converted into succinate, the 4 carbon molecule. Step 6 is the oxidation. Okay, so succinate undergo oxidation process, di mana dia akan remove the 2 hydrogen atom. Okay, then the 2 hydrogen atom dia akan pass to the FAD. So FAD is reduced to FADH2. Okay, so FAD reduced to FADH2, and the succinate, the 4 carbon is converted into fumarate 4 carbon. Okay, step yang ketujuh adalah hydration. Okay, so dalam step ni water molecule is added to fumarate to become malate. Then the last step yang ke-8 step 8 adalah proses oxidation. Okay, di mana malate is oxidized to oxaloacetate. Okay, the 4 carbon. So malate oxidized maksudnya dia akan berlakunya removal of 2 hydrogen atom. Okay, and the 2 hydrogen atom is accepted by the NAD+. So NAD plus is reduced to NADH plus H plus. Oxaloacetate is regenerated, and bind with the second acetyl enzyme A molecule. So the cycle will continue. So at the end of Krebs cycle, for one molecule of glucose, it will produce the 6 molecule of NADH, 2 molecule of FADH2, and 2 molecule of ATP. So di sini kalau lihat untuk one cycle for one molecule of acetyl CoA kat sini so yang kat sini adalah melibatkan satu molecule of acetyl CoA, it will produce berapa NADH. So dia akan produce 1, 2, 3 NADH. So bila dia pusing sekali lagi, dia akan hasilkan 6 molekul of NADH. Begitu juga dengan FADH2. Untuk 1 molecule of acetyl CoA, dia akan hasilkan 1 FADH2. So bila the cycle sekali lagi, so dia akan produce 2 molecule of FADH2. Dan juga 1 molekul ATP. Untuk 1 molekul of acetyl CoA. So bila the cycle sekali lagi, so dapatlah 2 molekul of ATP. So for the conclusion, actually dalam Krebs cycle ni dia akan producekan banyak the high energy molecule. So iaitu pada terdapat 2 NADH terhasil ataupun produce during oxidation of isocitrate into alpha ketoglutarate. And then 2 lagi NADH produced during oxidation of alpha ketoglutarate into succinyl coenzyme A. Begitu juga GTP is produced during conversion of succinyl coenzyme A into succinate, di mana 1 GTP ni dia akan transfer dia punya phosphate group to 1 molecule of ADP to synthesize ATP. So dia akan synthesize ATP ini secara substrate level phosphorylation. So dia akan hasilkan 2 ATP. And then FADH2 is produced during oxidation of succinate into fumarate. Dan 2 lagi NADH is produced, 2 lagi NADH produced during oxidation of malate into oxaloacetate, ataupun OAA. So NADH, so total ada 6 NADH dan juga 2 FADH2 ni dia akan enters the electron transport chain. So dia akan lalu electron carrier dalam electron transport chain. Di mana nanti dalam proses ni NADH ni dia dan juga FADH2 dia akan generate the ATP molecule juga. So nanti kita akan discuss dengan lebih detail lagi.